And so the next few slides will talk about how we identify chromatin states. And so the idea is that up to this point in the lecture, we've talked about epigenetic marks, like histone marks, DNA methylation, as well as chromatin accessibility. And so the idea of chromatin states is what people term the kind of recurring combinations of epigenetic marks across the genome. And so when we were talking about histone modifications, I mentioned that there's many combinations of histone modifications possible. And one important thing that people are trying to learn is, what are the most important combinations of histone modifications that occur, and where do they occur in the genome? And so chromatin states is basically the term people came up with to describe combinations of epigenetic marks. So those include both histone marks, as well as, for example, DNA methylation, as well as whether or not regions are accessible. So in a typical epigenomics assay, you typically assay more than one histone mark because, again, histone modifications have combinatorial effects. Sometimes their effect depends on other marks that are present on the genome. And so in this case, for example, here I'm showing you the results of assaying 5 different histone modifications, 2 of which are typically indicative of promoters, and 3 of which are typically indicative of silence regions. And you can see that already in this small example, what my data is telling me in this specific case is that both the STAT1 and STAT4 gene have basically active promoter marks near their start site, which then tells me that in this particular sample or in this particular set of cells, STAT1 and STAT4 are more likely to be transcribed than not. Similarly, in another region of the genome, at MYO1B, you can see that there's no significant promoter marks found near the start site, but you do see a lot of silencing marks throughout the gene body, as well as at the 5' and 3' end of the gene. And so this tells you that MYO1B is likely to be silenced in these cells. And so hopefully a recurring theme throughout this lecture so far is that histone modifications can mean different things depending on what other marks are also present on the same histones or nearby histones. And so here's another example where I'm showing you 5 different histone marks that have been profiled in a sample. And basically what you can see here is that certain regions of the genome, like CXCL14, for example, you can oftentimes see both kind of activating and repressing marks at the same time. And so here at CXCL1 and 4, you can see that K4 trimethylation as well as K27 trimethylation is observed at the transcription start site. And basically, as I've indicated here, K4 trimethylation is a mark of an active promoter, whereas K27 trimethylation is a mark of repression. And so this is an example of what's known as a Poise gene. So Poise genes, just like Poise enhancers, are elements that carry both activating and silencing marks at the same time. And so these regions are generally turned off, but they can be activated by removing, for example, the repressive mark. And so these kind of Poise genes are historically associated with genes related to lineage control and differentiation. And so again, basically the role of the K27 trimethylation is to repress the genes, but the role of the K4 trimethylation is to basically get them ready to be activated. So once the K27 trimethylation is removed, then the gene is ready to go at that point. So up to now, we've been talking mainly about a small set of 7 to 9 histone modifications that are frequently assayed, but it's worth noting that there are actually dozens and possibly more of histone modifications that are possible on all of the different NNC terminals of the histone proteins. And so what that essentially means is that in terms of all the possible combinations of histone modifications that could be found on a given nucleosome, you could have on the order of like 2 to the 50 possible combinations of histone modifications. And so if the effect of certain histone modifications depends on whether or not others are present, that means that there's a ton of possible combinations of histone modifications that could happen and, that might need to be thoroughly investigated to figure out what they do. And so what that basically means is that there's really a complex and possibly large number of combinations of histone modifications that could happen. And because histone modifications are dynamic, they're constantly being added or removed, it's really hard and really important to understand both what is the actual observed diversity of combinations of histone marks that I see in the genome, and what are their dynamics, like which combinations of histone marks might tend to follow others, and so on. And so an obvious question then becomes, well, how do we find meaningful combinations of histone modifications? How do we find the ones that happen the most often? And how do we figure out what they do? And so the classic answer to, how do you find combinations is a super common type of software called the hidden Markov model. Is frequently used to look at multiple histone modification assays and tell you basically what are the most frequently observed combinations and what are their dynamics in terms of which ones tend to follow which other ones on the genome and so on. And so hidden Markov models are a general kind of software that is used for many purposes. The two most kind of common software used to look at histone modification assays are called Chromate and Segway, and so you can look up those software tools if you're interested in actually running them. But basically on the next slide, I'm going to go over some very high-level basics about what kind of data goes into a hidden Markov model and what kind of data comes out of one. And so over the next few slides, I'm going to review what a hidden Markov model is and what it tells you about histone modifications across the genome. And so suppose in a hypothetical experiment, suppose we've done ChIP-seq experiments to assay the location of 3 different histone modifications across the genome. And so what I'm illustrating to you here are 3 hypothetical signal tracks that you would get from those ChIP-seq experiments, where basically the higher the green line is for a given histone modification, the more reads you saw at that particular region of the genome. And so you'll notice that across this hypothetical genome, I've divided the genome into different chromosome regions because typically hidden Markov models operate on whole regions of a genome, say like a 200 or 400 base pair window, instead of on the individual base pair level. And so hidden Markov model basically tries to answer 3 questions, number 1 of which is, across the genome, what combination of histone marks frequently co-occur the most? And it basically calls each unique combination of histone marks that you see a chromatin state. And so that's what I'll call output number 1 that we'll discuss in the next few slides. The second thing it tries to tell you is, given that it kind of learns or memorizes these frequently co-occurring combinations of histone marks, it tries to tell you where do these combinations happen. And so across the different chromosome regions that were delineated here by the dashed lines, HMMs try to tell you for each region which chromatin state or which combination of histone marks does it look like the most. And so that's what I'm calling output number 2 here. And so finally, it also tries to tell you something a little bit about the dynamics of these chromatin states. And so it tries to tell you, for example, if you are in one state of chromatin for one region, what is the likelihood that the next state over is of state type A or B or C or whatever. And so it tries to give you some idea about which combinations of histone marks tend to follow other combinations of histone marks, for example. And so that's what I'm calling output number 3. And so in terms of output number 1, recall that HMMs try to tell you something about chromatin states, and chromatin states are basically just unique combinations of histone marks that occur across the genome. And so in this hypothetical example, we've divided the genome into 7 different regions. And you can see that across these 7 different regions, even though we've assayed 3 different marks, and so in theory, you could have something like 2 to the 3 or 8 possible combinations of marks occurring across the genome. In this hypothetical genome, we actually only see 2 unique combinations of histone marks. So in one case, you can see that only K27 trimethylation is observed. So that seems to occur in chromosome regions 1, 2, 3, 6, and 7. And in the other case, you see K4 monomethylation and K27 acetylation. And you see that combination of marks in regions 4 and 5. And so hidden Markov model basically would look at these signal tracks and basically tell you that it saw 2 different unique chromatin states across the entire genome. Again, so the second thing that HMMs tell you are it basically tries to tell you for each region across the genome what is the state of the genome in terms of chromatin state at that position. And so basically for each one of these chromosome regions, the HMM intuitively looks at output number 1 and says, at this region, which combination of histone marks, which chromatin state do I see the most, is most likely here? And so in the table labeled output number 2, here I've just kind of labeled each chromosome region in terms of which chromatin state that region looks most similar to. And you can see intuitively that the states are labeled as basically AAA, BB, AA. And so one thing that's important to note here is that HMMs really only try to identify combinations of histone marks that co-occur and tell you where they happen across the genome. But by themselves, they can't tell you what those combinations of histone marks actually do yet. And so that's why here I've labeled the chromatin states hypothetically as just some state A and state B, just because at this point in time, we don't actually know per se what those states do. And so we can't label them as, say, for example, like an enhancer or promoter or transcribed region or something like that. And so the last thing that the HMM tells you is it basically gives you as output something called a transition matrix. And so the idea for the transition matrix is that if the HMM tells you that there are 2 states in this genome, it's going to give you a 2x2 table. And if there are, say, for example, 5 states, it's going to give you a 5x5 table. And so what this table tells you is that it basically, again, tells you if you are in one state, say state A, then it tells you what is the probability that the next state in the next region over is either going to be a state A or a state B region. And so, for example, in the top right-hand corner of this 2x2 table, the value that you see in that table tells you basically if a given region is in state A, what is the probability that the next region over is going to be a state B. And so let's walk through a hypothetical example of how you would actually fill out this transition matrix table. So in this hypothetical genome, we again see that the HMM called the states of this genome as AAA, BB, AA. And so let's consider, for example, let's identify all the different positions or regions across this genome that were labeled as A. And so you can see from output number 2 that regions 1, 2, 3, 6, and 7 are labeled as chromatin state A, or most likely to be chromatin state A. And so again, because this transition matrix tries to tell you if you're in, for the top row anyways, it tries to tell you if you are in state A, what is the probability of the next state over being an A or B? Then the way that you can fill out this table is that you can say, okay, well, I see chromatin state A in positions or in regions 1, 2, 3, 6, and 7. Let's actually just count how many times I see each state in the next position over in this genome. And so if we look at chromosome region 1, we see that we're in state A, and that's followed by another state A in region 2. And if you're looking at chromosome region 2, which is, of course, a state A, the next region over, which is in region 3, that's also an A, and so on and so on. And so what you'd see is that basically for chromosome regions 1, 2, and 6, basically those regions are of state A, and the next state over is also an A. Whereas if you look at region number 3 is called as state A, but the next one over is actually a state B. And the final region that you can look at is number 7, which is an A, but there's nothing after, because chromosome region 7 is the end of the chromosome, and so that state number 7, or the region number 7, can't contribute to this transition matrix calculation that we're making. And so if you look at the annotation over output number 2, what you would see is that of the regions that we're looking at, so regions 1, 2, 3, and 6, 3 out of 4 of those regions were followed by an A, and only 1 out of 4 of those regions is followed by B. And so in our transition matrix table, conceptually, the entry represented by the first row and first column would be a 3 out of 4, because 3 out of 4 of the times we saw an A followed by an A. And the top right-hand box would be a 1 out of 4, because in 1 out of 4 of the cases, we actually saw B following an A. And so in a similar way, we can fill out the bottom row of this transition matrix by looking at output number 2 and looking for all of the regions that are labeled as state B. And so in those cases, those are regions number 4 and 5. And so for regions 4 and 5, we can see that in one case, for region number 4, the B state was followed by another B. Whereas for region number 5, the state B there was followed by an A in region 6. And so in half the time, we saw a B followed by a B, and in half the time we saw a B followed by an A. And so in the bottom row of this transition matrix, we're going to fill out those entries as half and half. And so the transition matrix overall can be filled out by using the same kind of logic, and just kind of asking for every state, what's the probability of seeing every other state in the next position. And so here's an actual example from one of the first papers to use hidden Markov models to learn chromatin states on several different cell types. And so in this diagram, I'm showing you the signal tracks of different histone modifications that were measured in this case, 4 different cell types: ES cells, GM12878, HUVEC, and NHLF. And what I'm showing you on the bottom of this diagram are basically the called chromatin states for each region of this particular part of the genome that covers the WLS gene. And so broadly speaking, what you can see is that, and on top of these signal tracks, I'm showing you the structure of the WLS gene. And so what you can see is that, broadly speaking, the histone marks are the signal tracks that you see for the HUVEC and the NHLF cell types, so the 2 cell types on the right, have pretty different signal tracks compared to what you see in the ES and the GM12878 cells on the left. And so part of what I'm trying to illustrate with this slide is how you can actually interpret what you see from these chromatin states that you get out of an HMM. And so the point here is that suppose that in addition to these histone mark ChIP-seq profiles that you've collected, suppose that you've also collected RNA-seq data that tells you how highly expressed is the WLS gene in each of these 4 cell types. And suppose that your RNA-seq data told you that WLS is only expressed in HUVEC and NHLF. And so how that helps you kind of interpret what these chromatin states or these histone mark combinations tell you is that if you know that WLS is expressed in the right 2 cell types, but not the left 2, and furthermore, you can see that for the right 2 cell types, for HUVEC and NHLF, you can see that the chromatin states that overlap the WLS gene body, you can see that there's a lot of green, light green, dark green, and orange and yellow, but you don't see that very often on the cell types on the left for WLS. Then what you can kind of infer from that is that the dark green and the light green probably correspond to something like transcribed regions of the genome. And you know this because WLS is only expressed in the right 2 cell types and not the left 2. And those green chromatin states are overlapping that gene body on the 2 right cell types, but not the left 2 cell types. And so basically the point here is that when you combine these like histone mark ChIP-seq profiles into chromatin states, and then you combine that with like RNA-seq data and kind of knowledge of the locations of the genomes, that can help you start to understand what do these histone mark combinations tell you. And so the other thing that you can also see is that near the transcription start site of the WLS gene in HUVEC and NHLF, you can see a little red chromatin state indicated that basically overlaps right where the TSS starts. And so because you know that the WLS gene is being transcribed in those right two cell types but not the left two, you can further infer that the chromatin state represented by those red boxes probably suggests that those are like active TSS marks, for example. And so finally, what you'll also notice is the kind of open red box that I've drawn on the HUVEC diagram. That indicates a region of the genome where you see a lot of H3K4ME1 and H3K27 acetylation marks. And you don't see that as much in the NHLF cell type. And so you can kind of infer from the fact that, maybe you knew ahead of time that K27 acetylation is a mark of strong enhancers, but you may not have known what the other marks do. From this knowledge, you might guess, for example, that region represented by the open red box might represent something like a cell type specific strong enhancer, because that K27 acetylation mark and the other marks associated with it are kind of very strongly present in the HUVEC cell type, but not the NHLF cell type. And so here's a summary of the different epigenetic marks that are associated with each different chromatin state from that Roadmap Epigenomics project. And so it's worth pointing out that the hidden Markov model will take data from different epigenetic assays and chromatin accessibility assays and tell you what are the combinations of epigenetic marks and accessibility are most frequent across the genome. They'll tell you, what the transition matrix are. They'll tell you where each state is plausibly located in the genome. But they won't actually assign like functional labels to those chromatin states. It won't tell you ahead of time, okay, like this combination of marks basically represents active transcription start site. This combination of marks represent like enhancers or bivalent enhancers and so on. Those kind of annotations, are left to you to figure out. And so, just by looking at the combinations of marks alone by themselves is, it's not really possible to identify what is the functional effect of those given chromatin states. And so oftentimes you have to look at these, the co-occurrence of these marks along with other kinds of data like RNA sequencing data that we'll talk about in the transcriptomics lecture, or you might have to look at like POL2 binding or CTCF binding or things like this to really, or things like gene annotation where you, for example, might know where the existing coding sequences are, or you might know where existing enhancers or promoters are, in order to really kind of say, okay, well, these states tend to occur where I see protein coding genes, and so these states probably correspond to either active transcription or active repression of a gene body, whereas these other states tend to co-occur where I know there's functional enhancers, and so these states might represent marks associated with active enhancers. And so some of the take-home messages from this slide are that number one, you can see that DNA accessibility is kind of broadly anti-correlated with DNA methylation, which is kind of what you'd expect. So generally speaking, we said at the DNA methylation part of the lecture that DNA methylation is broadly associated with repression of gene expression, at least outside of gene bodies. And so you can see that here, in that the active transcription start sites, for example, have very low levels of DNA methylation, but high levels of DNA accessibility. And on the other hand, the repressed polycomb-associated regions are associated with higher levels of DNA methylation, and as well as the kind of quiescent or low activity regions of the genome. Those are also associated with high levels of methylation and low levels of DNA accessibility. And so the exception to this rule are gene bodies.
where you can see that strong transcription tends to be associated with high levels of DNA methylation as well. And so DNA methylation tends to also mark actively transcribed gene bodies. And so one of the other kind of major take-home messages from this slide is that basically you can see by looking at the COVE column, which represents coverage across the genome, average coverage across these 113 different cell types, you can see that only about 20% of the genome seems to really encode recognizable, active transcriptionally associated elements like promoters, enhancers, and transcribed regions. And the rest of the 80% seems to either be like repressed, mostly repressed or quiescent or low activity, low or no functional activity across the genome. And so in short, 80% of the genome not doing anything transcriptionally active is pretty large proportion. And so one of the key open challenges in human genomics and genetics today still is to kind of understand, what is that 80% of the genome that's currently quiet? What is it actually doing? Like, is it actually devoid of true function, or is it just doing something that we don't currently know about or we don't currently recognize?